Hello everyone. Let's get started. Today we're going to be talking about universal characteristics of cells and cellular biochemistry. We can define a cell as being the membrane-bound subunit of all living things. So if an organism is alive, and if it's living on our planet, it is composed of one or more of these subunits called cells. Now, the discovery and description of cells has a very interesting history, and there are a couple of historical names that you should be familiar with. The first name is Robert Hooke, and the second is Antoine van Leeuwenhoek. Both of these men did their research in the mid-17th century, and both of them contributed to our very early understanding of what the subunit of living things, or the cell, is. Robert Hooke is credited with being the first person to name this subunit as a cell. It was Hooke who took a very primitive light microscope capable of about a 30x magnification, and examined a sample of cork tree bark. Under his microscope, this bark looked like it was composed of many smaller compartments, and it reminded Hooke of little rooms that you might see inside of a monastery. Remember, a monastery is a place where monks live, religious monks, and they are typically assigned their own room, their own small and sparse room, that is referred to as a cell. So Hooke decided that these little compartments that he was seeing inside of the cork bark sample could also be called cells, after these small rooms. Now, Leeuwenhoek was interested in Hooke's work and took it a step further. Leeuwenhoek was able to advance on the primitive form of a microscope that Hooke was using. He was actually able to develop a microscope that was capable of about a 300x magnification using better lenses. And what Leeuwenhoek did was examine not just plant cells, but all different kinds of cells underneath his microscope. He's credited with describing blood cells, sperm cells, some algae that he collected, and some bacteria from the environment. Now, Hooke and Leeuwenhoek were not the only scientists who were working on discovering and describing these subunits called cells. Many other scientists were working at the same time, and throughout the 17th, 18th, and even into the 19th centuries, all of these scientists together formed what we now refer to as the cell theory. The cell theory has two parts. The first statement says that all organisms are composed of cells. The second statement says that all cells come from pre-existing cells. So again, if it's a living organism on our planet, it's composed of one or more of these subunits called cells. All organisms are composed of cells. And cells don't appear out of thin air. Cells come from other cells that go through the process of cell division, with one parent cell dividing into two daughter cells. So all cells come from previously existing cells through, again, this process called cell division. When you look at all of the living things on our planet, you can again describe them as being composed of either one or more cells. So we use terms to categorize living things based on whether they are single-celled, multicelled, and what kind of characteristics those cells have. Remember, this term prokaryote refers to the type of organism that is single-celled, and within those cells there are no organelles and there is no true cytoskeleton inside of the cell. Those are the prokaryotes. Now, eukaryotes are quite different. Eukaryotes can be single-celled, but they can also be multicelled. Eukaryotes contain organelles inside of their cells, and they also contain a true cytoskeleton. Now, the terms prokaryote and eukaryote are certainly not the only terms we use to categorize living things. We also categorize living things based upon levels of taxonomy, and you've probably heard of these before. The most recent grouping, the most broad grouping in taxonomy is what we call the domain. So starting with the domain, we can then describe organisms as belonging to a kingdom, and within the kingdom, a phylum, within phylum, a class, and then an order, a family, and finally a genus and a species. The three domains of living things on planet Earth are the bacteria, which evolved first, the archaea, which evolved next, and the eukarya. Now, in this diagram, we're looking at a phylogenetic tree. And if you remember from your evolution class, the base of the tree, or the base of the trunk of the tree, if you will, this represents the universal common ancestor. This is the ancestral form of life that all living things on planet Earth share. Now, if we travel up the tree, we hit a branching event. We hit what we call a node. Something happened at this moment in our ancient planetary history that split the living things on the planet into two different groups. Some of those organisms went on to become the bacteria, and some of them traveled on to eventually become the archaea. But there was another important critical split that occurred here on the tree at this other node, and that event split the archaea again from what would become the eukarya. You'll notice that within the domain of eukarya, there are 4 different kingdoms listed: Animalia, Protista, Plantae, and Fungi. So animals, plants, protists, and fungi. The bacteria and the archaea are prokaryotes, and the eukarya are, as the name suggests, eukaryotes. Now, the domains are important for us to understand in a course like this. I'm not going to test you on the other designations in taxonomy, but if you're curious, we can use these designations to better understand human beings. If you look at this list down here, humans are, of course, eukaryotes, so our domain is eukarya. We belong to the kingdom Animalia, the phylum Chordata, the class Mammalia, we are mammals, the order Primate, the family Hominidae, the genus Homo, and the species Sapiens. All living things on this planet can be classified using this system of taxonomy, which means all living things have a genus and a species. Now, one of the most important things that we have to understand as we start our journey into the study of cell biology is we have to start to develop an understanding of the size and the shape of cells. Cells vary tremendously in their size. On this slide, you're looking at a scale that ranges all the way from 0.1 angstroms on the far left all the way up to a centimeter on the far right. And along this scale, you can see where different types of cells have been placed. Now, there are other things on this scale besides cells. There are what we would call small molecules. Down towards the lowest end, there are viral particles, and then there are cellular forms of life. Now, there are no archaea drawn on this scale, but generally the archaea and the bacteria are approximately the same size, and that size falls in the range of single micrometers. If you then compare that prokaryotic type of cell, to a eukaryotic type of cell, you can see that animal cells are about 10 times bigger than prokaryotic cells, bacteria and archaea. So if you had a bacterium, in other words, that was a micrometer in diameter, the animal cell would be a full 10 micrometers approximately in comparison. If you compare animal cells and plant cells are a further 10 times larger. So again, in general, plant cells are much larger than animal cells, and animal cells are much larger than prokaryotic cells, bacteria and archaea. Viral particles, it's important to note, are not cells. They do not meet a definition that we're going to discuss for cellular life. Viral particles are not technically alive. They simply do not meet the definition for a living thing. You'll notice that they are significantly smaller than the smallest types of cells. Viruses tend to range in the nanometer, 10 to 100 nanometer range. And of course, if you compare viral particles to some of the small molecules within cells, we're talking a factor of 10 or a factor of 100 smaller. The other thing you should note about this diagram is that while cellular life is visible under a light microscope, viral particles and molecules are not. It means that even under the highest power lens, the 100x or the oil immersion lens, using a light microscope, you cannot see viral particles or any kind of molecule. Now, just as cells vary tremendously in size, cells also vary tremendously in shape. And it's important to know that the shape of a cell is tightly related to its function. Form and function go together. If you look at the picture on the left-hand side of the slide, what you're looking at are drawings of a number of different cells. You can see a red blood cell up here on the upper left. Next to that, there are 3 muscle fibers drawn together. There's a neuron. There's a paramecium, a single-celled eukaryotic organism down at the bottom. There's a diatom and also a plant cell, or a group of plant cells, making up a pore on a leaf. Just by looking at these drawings, you can see that there's a tremendous variation in the physical shape of living cells. And the shape of those cells matters. It's a critical component of how the cells are able to function. So, for example, if you're a red blood cell and your function is to both carry oxygen to tissues, exchange that oxygen for carbon dioxide, and then carry the carbon dioxide away from tissues, It's critically important that you have as much surface area to your membrane as possible. And red blood cells achieve this by having this very unique sort of a shape. They are certainly circular, they're round, but they have a depression in the center that increases the surface area in that region of the cell. If you look at a muscle cell, and we call muscle cells muscle fibers, and you think about the function of a muscle fiber, you know that for skeletal muscle at least, that function is contraction. The job of the cell is to get shorter, contract, and then get longer, relax. It wouldn't make sense for a muscle fiber to be circular the way a red blood cell is. Instead, they are quite elongated, and that's why we call them fibers. As this elongated cell contracts, it literally shortens in its length. And as it relaxes, it gets longer. Form relates to function. All of these strange sort of alien-looking projections coming off of this neuron represent what we call dendrites, and dendrites are simply projections of the membrane. The job or the function of this neuron is to gather information and relay it to the nervous system. All of these dendrites come in contact either with the environment or with other neurons, and information is exchanged at these points of contact. Form follows function. Over here on the right-hand side of the slide, you're looking at one here and then one here, two animal cells. The junction between the cells is right here. These are animal cells that are being examined underneath an electron microscope, so much more powerful than a light microscope. And if you recall from the previous slide, electron microscopes are capable of magnifying even the very smallest parts of a cell. So under an electron microscope, we are able to see some of the very specific structural components of these animal cells. These cells happen to be the kinds of cells that line the intestine. And as you can see, they have these very regular, very elongated, finger-like projections of membrane coming off the surface. These are called microvilli. And just like with the red blood cell, the microvilli of intestinal epithelium serves a purpose. It increases the surface area of the membrane of these cells. And the job that these cells are trying to perform is the absorption of nutrients from the intestine. So by increasing the surface area with these microvilli, these cells are better able to do their function, their job. Now, when we compare one type of cell to another, they can look very different. They function very different. When you compare prokaryotes and eukaryotes, there are lots of differences. But there are certain things that all cells have in common. There are what we refer to as universal characteristics of cells, and I have 7 of them listed here that you should be familiar with. The first and most obvious universal characteristic of a cell is the fact that it is surrounded by a membrane. And remember, we call that a cell membrane, a plasma membrane, or just a bilayer. All those terms mean the same thing. It's the surrounding structure of a cell. And all cells have a membrane. All cells store their genetic information in the form of DNA. All cells use what we call semiconservative replication to copy that DNA at the time of cell division. All cells express their genes in the DNA. They transcribe the DNA into RNA, and they translate the RNA to produce protein. Remember, it's the proteins that living cells produce that give them their unique traits. All cells use certain types of proteins as catalysts to increase the rate of biological reactions. And remember, we call these biological catalysts enzymes. And finally, all cells contain 4 distinct macromolecules. These are the building blocks of all of the structures within the cell: proteins, carbohydrates, lipids, and nucleic acids. Now, at the bottom of the slide, you can also see another list, the list of the 4 universal structures that you find inside cells. So again, all cells have a membrane, all cells contain cytoplasm, all cells contain DNA, and all cells contain ribosomes. Remember, only eukaryotic cells have organelles, so that tells you something. That tells you that ribosomes are not technically organelles. Ribosomes are found in all cells, both prokaryotic and eukaryotic. Now, an interesting question that has been asked throughout history is the question of where cells came from. Where did the original cellular life form come from on the planet? And scientists have been researching this question for decades, if not centuries. There's an important group of scientists and an important experiment that you should be familiar with in terms of the history of the emergence of cellular life. It's an experiment that took place in the 1950s by a couple of scientists named Miller and Urey. So we refer to this as the Miller-Urey Experiment. And it's the experiment that showed us this process that we now refer to as chemical evolution. What Miller and Urey demonstrated in this experiment is that if you can replicate the very conditions that were present on the Earth many billions of years ago, you can demonstrate the production of the sorts of small and medium-sized molecules that would be necessary to produce life, to produce cellular life. Miller and Urey set up this very complex-looking apparatus in their laboratory, and they combined, again, the elements that would have been present on the very young Earth. So they combined water with heat and light. They added in the sorts of gases that would have been present on the planet at that time, things like hydrogen gas and methane and ammonia. They heated those gases up, again in the presence of water, of steam, and then they applied electricity, basically sparks of electricity, which would have been present in the primitive atmosphere around the planet. And what they demonstrated was that this combination of features was able to produce some very, again, small and medium-sized organic molecules, carbon-containing molecules that could have then gone on to combine with each other as the Earth continued to mature and produce living things. Now, lots of other work has come into play since the time of Miller and Urey's famous experiment, but just recently, another experiment was published that added to our understanding of where cellular life came from on our planet. This experiment is referred to now as the Sutherland experiment. This work was published in 2009, and it basically expanded on the work that Miller and Urey did. The Sutherland experiment showed us how RNA could have first appeared on Earth. And if you recall, RNA is the form of nucleic acid that we believe emerged first on the planet. So RNA would have been the genetic material that was inside of the very first primitive cells on our planet. So it was Miller and Urey who showed us the possibility of chemical evolution producing these small and medium and even larger molecules. Sutherland's group then showed how those small, medium, and larger molecules could have combined to produce some of the bases that are found in RNA. Basically, what they did was they set up a fairly similar apparatus to Miller and Urey's, but they added in what we believe are these partial components that you would need to build RNA. So basically, they put in partial ribonucleotides, particularly partial cytosine. So pieces of what could come together and become cytosine or C in RNA. And by, again, applying heat and light and electricity to a watery environment, they were able to show that partial ribonucleotides could combine with some of the other small and medium-sized molecules to complete cytosine, to complete one of the ribonucleotides. And they also showed, interestingly enough, that ultraviolet exposure under those experimental conditions can convert cytosine into guanine. So what the Sutherland experiment did was show in the laboratory that it was possible for both cytosine and guanine to emerge on our very early planet. Very interesting experiment. If you're interested in learning a little bit more about the Sutherland experiment, I would direct you to YouTube to an episode of Nova called Nova Science Now. And the episode was called Where Did We Come From? I have the link down here. Again, if you'd like to take a look at a short video, you can learn a little bit more about these famous, groundbreaking experiments. So while we are still gathering experimental information about where the first cell came from, we're well on our way to understanding how it could have happened on our early planet. We now believe that the first life form that emerged on planet Earth was not actually a cell. In other words, cellular life evolved from a more primitive form of life, a very primitive form of life, what would have been a self-replicating RNA molecule. Remember, we believe that RNA was the first nucleic acid to evolve, and DNA emerged out of RNA. So, knowing that the components of RNA could have evolved through chemical evolution, our current belief is that RNA emerged outside of a cellular environment. And it was a kind of RNA that's called a ribozyme. Now, this would have happened greater than 4 billion years ago. Okay, so back before the first cells appeared on the planet. This self-replicating RNA had that one primary characteristic that we demand that all living things have, which is that they can reproduce themselves. So that's about all this RNA was able to do. It was able to make copies of itself. And again, that's what we call a ribozyme. The first cell would have come much later. Events would have taken place to most likely enclose that ribozyme inside of a membrane. And of course, that would have happened somewhere around 3.5 to 4 billion years ago, which is when we believe the first cells appeared. In this drawing down at the bottom, you're looking at an artist's rendering, again, of a very primitive cell. It's basically a very simple bilayer of membrane with RNA inside of it, and just a few of the molecules that we would now expect to find inside of the cytoplasm of a modern cell.